Good morning, students. Today we are going to cover the topic lipids. This is your lecture number 3 for the course name Biology for Engineers, and the course code is BBS01T1008. So after the other biomolecules, I have covered carbohydrate in the previous class. Lipid is the next biomolecule we will be discussing in detail. So recapitulation that you should be known for, that is the previous classes we have covered with the cell membrane and the plasma membrane, the cell composition and its functions, and the macromolecular structure, shape, and the other detail about carbohydrate. So after this all recapitulation, you will be better able to understand lipids in detail. So firstly, we should talk about what are lipids. So lipids are macromolecules made up of fatty acid monomers, and the function of lipid includes the structural support for the cell, energy storage, cell signaling. Lipids are non-polar in nature and do not interact with the water. You must be remembering that in the phospholipid bilayer, specifically if I'll take one of the phospholipid composition, I can see this is hydrophilic head and the hydrophobic tail. The hydrophobic tail, which is of fatty acids, and the triglycerides can also be seen here in the head and tail like orientation. Steroid and waxes. So this is the structural composition of steroid and waxes that I'll discuss in detail later. Currently, you need to know what is the composition of these particular lipids. So I hope everybody can understand. This is the fatty acid tail composition present in the phospholipid, and this is hydrophobic in nature, whereas the head orientation is hydrophilic in nature within the phospholipids. So the types of phospholipids or lipids I'm talking about, one is phospholipid, triglycerides, and steroid or waxes are coming under the same category of lipids. So now we will talk about the basic introduction in detail about the lipids. So lipid are one class of the large biomolecules that do not form the polymer. Like in the carbohydrate, we have studied the monomers do combine to form the polymers like polysaccharides. But lipids are one of the class of large biomolecules that do not form the polymers. The general feature of lipid is having little or no affinity for water. That means it is not going to be soluble in water. Lipids are hydrophobic because they have mostly hydrocarbons which form non-polar covalent bonds. That is why it is insoluble in water. So most biologically important lipids are fats, phospholipids, and steroids. So if I talk about fats, fats are constructed from two types of smaller molecules which are glycerol and fatty acids. Like we have seen in the phospholipid bilayer, the tail portion is made up of fatty acids. So the fats are actually having two smaller molecules, one is glycerol, another is fatty acids. The glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. So, and the fatty acid consists of the carboxylic group attached to the long carbon skeleton. So I'll be showing you the diagram related to these particular lipid molecules. Like I have shown you in the steroid diagram in the previous slide, I'll be discussing here in detail. So this is the cholesterol composition. If I talk about a free fatty acid chain, so we can see here. This is the free fatty acid chain, a triglyceride. This is a very complex macromolecule which is biologically active, present in the plasma membrane. This is a triglyceride. All are the example of lipids. And the phospholipid can be seen here. Structurally, it is very complex. So to simplify that complex structure, I just need the diagrammatic presentation how lipids are present. I hope everybody can understand this phospholipid bilayer that we have studied in the cell membrane. So each cell is surrounded by a cell membrane, and the cell membrane is having phospholipid bilayer like this. So you can see the structure here. I am going to talk about this lipid composition by taking the same phospholipid bilayer. This is the head orientation, which is hydrophilic, and this is the tail orientation made up of fatty acid, and this is hydrophobic. So you can see this is one strand of the lipid layer. You can see here I have tried to show the saturated lipids only. So saturated lipids are looking like so. If I'm talking about mixed kind of lipids, unsaturated and saturated or monosaturated, then if monosaturated lipid is taken, you can see this is the strand is shown here like this. So if I'll talk about how it is going to be, in the cell it is going to be performing its activity or cell exchange within the cell membrane. So that you can see this is water-like orientation. Water is present near and around the head part, which is hydrophilic polar head, whereas the tails are non-polar. Tails are non-polar. These are hydrophobic. So water orientation is just found around the hydrophilic polar heads. So lipid is a macrobiomolecule that is insoluble in water but soluble in non-polar solvents like organic solvents. So non-polar solvents are typically hydrocarbons, a category which is organic, having hydrocarbons, used to dissolve in naturally occurring hydrocarbons, lipid molecules, but that do not easily dissolve in water. That means the fatty acids are insoluble in water, they are hydrophobic in nature. So all fatty acids, all waxes, steroids coming under the category of lipids are going to be water insoluble but fat soluble or organic compound soluble. So if I'll categorize which are soluble in hydrocarbons and organic solvents, that will be, you know, that will be lipids. But if I'll further classify them that do not dissolve in the water, including fatty acid, waxes, steroids, but fat soluble, vitamin A, vitamin D, vitamin E, and vitamin K. Monoglycerides, diglycerides, triglycerides, and phospholipids all are fat soluble. So we need to categorize it water soluble area and portion and the fat soluble portion. Correct. So fat soluble compound also are monoglyceride, diglyceride and triglyceride, whereas water soluble is only the head part of any phospholipid I'll be taking for. The function of lipid includes the storing energy and the cell signaling. It is providing a selectivity or membrane selectivity action by its thick lipid composition present within the cell membrane. So the function of lipid is to store the energy and to provide signal to the cell and acting the structural components of the cell membrane, like I told you, as head and tail orientation. So the lipids have application in the cosmetic and food industry as well, and currently it is having application in nanotechnology profoundly. So if I'll go deep into the lipids, how they are available, how they are naturally present, how they are soluble, and the further detail of lipids is going to be lipids are hydrophobic and or amphiphilic small molecules. The amphiphilic nature of some lipid allow them to form the structure in the form of vesicles, which is multilaminar, and the membrane in the aqueous environment, because half of its part, especially head part, is generally in the phospholipid head part, it's going to be soluble in water. Biological lipids, if I'll be talking about, these originate entirely or in parts in two distinct types. So biochemical subunits or the building blocks are ketoacyl and isoprene groups. Lipids may be divided into 8 categories. One is fatty acid, another is glycerolipids, glycerophospholipids, sphingolipids, and this is saccharides, polyketides. These are the lipids derived from the condensation of ketoacyl subunits, and the sterol lipids and phenol lipids derived from the condensation of the isoprenes of lipids. So lipid is sometimes used as a synonym for the fats. Generally we are calling it as fats. And fats are the subgroup of lipids which are called as triglycerides. The triglycerides also enclosed or encompasses the molecules like fatty acids and their further derivatives. It may be triglyceride, monoglyceride, diglycerides, and phospholipids. So cholesterol is also coming under the same category. So it is about the detail of nature and the composition present within the nature, composition and types present within the lipids. Now we should talk about the structural composition of the lipids. So this is the long fatty acid chain. This is, you can see, palmitic acid. This is long fatty acid chain. Acidic orientation is at one end, and this is made up of hydrocarbons. You can see. So fatty acid chain, if you are talking about, this is going to be like so. It may be any long depending upon the fatty acid you have taken. So here currently I have shown for palmitic acid. So you can see one orientation is having acetic group. If I will talk about the glycerol, like I have shown you in the beginning, it is having OH group at one side and H group on the other side. This is 3 carbon long chain. So the dehydration reaction in the synthesis of fat. Now we are talking about the ester linkage is present here. So by the dehydration reaction in the synthesis of the fat, so this is the fatty acid and the glycerol. Now we will be going for dehydration reaction in this particular. So you can see here the H and OH, H from glycerol and OH from lipids are going to come out as dehydration reaction, and you can find here a kind of ester linkage. So this is the ester linkage with one chain of fatty acid. This is the another ester linkage with another fatty acid chain, and the third ester linkage in the fatty acid chain. So this is called triacyl glycerol. So the lipid and category of lipid, I have taken how it can be derived or it can be synthesized. Fat molecule which is triacyl glycerol can be prepared by the dehydration reaction in the synthesis of fat. So again we can go through. This is the glycerol molecule and this is the fatty acid long chain, palmitic acid. So it is one part of the reaction that we have already gone for, and this is the other part of the reaction which is obtained as a result of dehydration reaction. This particular is having lot of ester linkages depending upon, like glycerol is taken here along with the fatty acid chain. So 3 ester linkages are found in the fat molecule, which is also named triacyl glycerol. Now we will talk about the further types of the lipids. So fats separate from the water because of the water molecule uh, form a hydrogen bond with each other and exclude the fats. In fat, generally 3 fatty acids are joined to the glycerol by an ester linkage. We have seen in the diagram also, creating or forming a triacyl glycerol composition, or you can call it as triglyceride. So the fatty acids are very long. It is a network of hydrocarbon long chain, and the number as well as the location of the double bond are defined as per the type of the fatty acid is taken there for the reaction. It can be divided into 2 categories or 2 types. One is saturated fatty acids, another is unsaturated fatty acids. Like in the diagram in the beginning, I have shown you the unsaturated and saturated fatty acids. So saturated fatty acids are those have maximum number of hydrogen atoms possible and no double bonds are present. By name it is clear that it is saturated, having single bond only. Unsaturated fatty acids having one or more double bonds present within the lipids. You can see the diagram here to get to know it clear. Here I have shown you this is a triglyceride composition. That time I have shown you with the carbon chain. Here I have shown you with the skeletal orientation with glycerol. So you can see here this is ester linkage. That is why it is called as triglyceride. So the structural formula of the saturated fat molecule, you are not finding any double bond here in this whole triglyceride orientation. It may be stearic acid, it may be palmitic acid with which you can react the glycerol by dehydration process or reaction, and then you will get to have the number of ester linkages. So if it is having saturated orientation, then it is going to be called as saturated fat altogether. It may be through stearic acid or it may be through palmitic acid. Then I'll go to another structural formula of unsaturated fat molecule. You can see here this, the long chain fatty acid which is having double bond present. So it may be cis double bond present here and forming the stearic acid chain. So I'll be going what is the stearic acid and palmitic acid chain looking like. Structural formula of a saturated fat molecule is taken here. Simply you can see here fat, that means here I have taken the example of butter. You can see this is a saturated long chain of fatty acids, and stearic acid is a saturated fatty acid having all long chains having single bond present with it, like I have shown in the previous diagram. But if we'll go for this unsaturated fatty acid, you will be finding double bond present. So cis double bond causes bending of this molecule as well. Earlier it was a straight long chain when it was saturated, but once it is getting unsaturated, the chain may also cause bending within. So the lipid orientation may be of saturated kind and unsaturated kind can be studied here. Now for references, you can go through certain references of the journals. Journal of Lipid. For the diagrammatic help, to make it very clear to you, I have taken a video for reference as well as Pearson's Educations 2008 for diagrammatic presentation as help to make this presentation much lively and understandable. So this is for the whole lecture. I hope you understood it well. Thank you.